Welcome to this ReadStore tutorial where we will look at the project and dataset management capabilities of ReadStore. This is really at the core of the functionalities because it gives you the option to nicely structure your NGS datasets, your projects, and then also later take these structure to your analysis to make your analysis workflows more automated and easier to reproduce. What we'll first look into in this video is a quick start tour of how we can create a project and assign datasets. And then in the second part of this video, we will take a very detailed look into the different options for creating the projects, datasets, and how they are structured. Okay, so we start in the datasets overview here, where we get a list of all the NGS FASTQ datasets that we have uploaded. How we're doing the uploads I showed on a previous video, which I'm going to link. You can have a look how the upload and staging procedure works. And here we have now 6 datasets, 3 replicates from a tumor and 3 from a normal tissue. And we can already see that they have not yet been assigned to a group. We also have some more information on these datasets, for instance, if they passed QC or if they are paired end. And what we're first going to do is create a project. For this, we move to the projects page here and simply click the create button. We don't have any projects created, so this field is all empty. And in the create projects dialog, we start by entering a name, and we call this the cancer project. And we will add a little description. And finally, what we want to add is some metadata. So metadata are really important because they are key value pairs that allow you to make a structured access to these datasets later, and they can also be imported in your analysis scripts through the command line interface or the SDK in Python and R scripts, and this can help you later to guide your analysis. So for instance here, what we will do is we're going to add as a key here a cost center, and this helps us to later also group our datasets and search them, for instance, by these different key value pairs. This is very useful and it's a very important feature. Then we're going to set these dataset keys. These dataset keys are also keys that are then becoming the metadata keys of all the datasets that are associated with this. So this is typically something like a replicate or the tissue type. So basically anything that can help us to identify and group the datasets that are associated with. And we can also edit this later in our datasets, but here it really helps us because it will be pre-filled in the dataset. So now we defined all the metadata here. Next, we can assign some datasets. And we have basically a list here of all the datasets that are available in our group to be assigned. We can just select all of these and add them to the attached datasets. That's all. This will basically attach all these 6 datasets into this project. And finally, we have an attachment tab here. And this attachment tab is meant to store any type of files that you would want to archive along with your project. And this can, for instance, be a PowerPoint presentation. So we prepared here a PowerPoint presentation that's called Library QC. That is containing QC information on the datasets that are assigned to this project, for instance. This could, for instance, also be plots or any type of downstream analysis data that you would want to store along with your along with the NGS datasets. And we just hit the confirm button, and this is going to create this dataset now in our overview. And here we can then see that we created this cancer project with a description here. And if we click here on this project and if we have the details activated, we can get an overview of the project features here also in this details view. Again, this is some information about the project itself, the metadata, all the datasets that are attached here, attachments, which we can also download here via this button. And finally, we also have collaborators in case we share datasets between groups. And that's it. We can then go to the dataset section, and here we see that all these datasets now have a project attached. What we can do now here is update these data sets in case that we want to make use of the metadata. We see that the dataset keys that we created for the projects are now also available here in the dataset. So for instance, the replicate and the tissue. And we can, for instance, say this is replicate 3 and the tissue is tumor. And if we save this, then these metadata will be saved along with the datasets. Our dataset overview, we can also directly look into the metadata. For this, we can switch here the metadata toggle, and this will give us an overview of all our datasets in which the dataset ID and name are shown together with the dataset keys as column names and the values for each dataset listed here. This helps us to get an easier understanding of the structure of our datasets, and for instance we can group them here by the tissue type or replicate. Also, if we have more datasets than this single projects, we're going to switch back here and now take a look at the detail view. The detail view allows us to easily check individual descriptions of those datasets here. For instance, name, description, the group it's associated with, and when it was created. Here, this feature view also allows us to look at individual reads that are attached to these datasets, so the FASTQ files directly. For this, we can click here and then we get an overview of the quality, which is automatically calculated by ReadStore, and important features, for instance, the read length, the number of reads attached to this dataset, the size, and if we scroll down, we also have the option to download these FASTQ files from our browser if they're not too large. And if we go back, we get an overview of all the projects that the dataset is associated with. It can be associated to several projects, and then also a list of attachments or files that we assigned on the dataset level, which is also possible. Finally, there is an export function here that allows us to export this view as a CSV file and import it again in Excel, for instance. And those are the basic functions in this dataset overview. So let's have a look at the project and dataset structure, and we will start by looking at the datasets. So datasets are composed of different read files, metadata, and attachments. And most important are, of course, the read files. Those are the FASTQ files, along with additional information on these FASTQ files. For instance, the path from where they were uploaded, the number of reads, the size, the read length, quality metrics. And depending on the assay we perform, depending on whether it's paired-end sequencing or if we want to store index reads, there are different numbers of FASTQ files attached to each dataset. And along with the FASTQ files, we can save some metadata. We've seen those are key value pairs that help us to later structure our datasets and group the data in a flexible manner. For instance, we can add replicate information. We can add information of the source of the data, for instance, a patient ID, assay information, anything can store in this key value format. And finally, there are attachments. Attachments are arbitrary files that we can store along with our dataset. So we also store plots or scripts from downstream analysis here in these attachment files. Next, we have the projects, and the projects are a structure that hold different datasets. This is meant to group datasets that belong together or that should be analyzed together into one logical unit. So a dataset can be part of different projects, and different projects can have multiple datasets. So this is very flexible. And projects also have metadata in a similar key value format for storing additional information. There's an option to add attachments, as we have seen, and there are dataset keys. And dataset keys are basically metadata keys that the datasets of the projects that are attached to projects inherit. And then we don't need to type this in anymore as metadata keys of the dataset. So this comes in handy if you want to structure your datasets and pre-fill metadata on the level of a project. Finally, we have different options and operations that we can run on the projects and data sets. So of course we can create and update them. We can share projects and this allows us to work in a cross-functional way. If we're an organization where we have different departments and where we have restricted file permissions, we can share projects and a few data sets of our inventory with collaborators from another group so they can access their data sets and projects. We can also export data set and project overviews. We can download the FASTQ files, and then of course we can delete those. And we will now look in a more detailed manner into these individual operations in our ReadStore app. So let's start with the create operation. We've actually already seen this in the first part of this video. We are now on the projects page, and if we want to create a project, we just press the create button, and then we can go through the procedure that we've seen in the beginning. We can enter name, description, we can modify our metadata, we can attach data sets here by selecting them, and finally we can also add attachments to this project. For creating data sets, it's actually very similar, but the creation of data sets is going through the upload and staging process. We've seen this in another video, so I'm not going to show this here. And once we created a data set or a project, we can select them and update them. And this again gives us access to the dialog here where we can edit individual attributes. So for instance, we can, for this data set, add another metadata key. So let's say we want to store some assay information over the update operation. And the same interface is then available for the projects, where we can also select our projects. Then the update button will become available. And here we can, for instance, confirm the update. This will be saved in the database. Next, we want to look at the share functionality, where we can collaborate on individual projects across teams and across departments, for instance. This is a feature of the advanced ReadStore versions that comes with a more elaborate user and group management. For this, we can select our project, and then we can click the share button. And the share button will give us access to a list of all users that are available within our organization. And we can select users that we want to share this project with. So in this case, we have Eva and Mike, and we select them as collaborators and then confirm this. And this means that they now see this project, although it's not part of their group, as part of their workspace, and they have read access to the attachments, to the data sets, and so on that are part of this project. So let's take a look at the export function. The export function allows us to take all data in our workspace and write them to a CSV file that we can, for instance, open in Excel. And it will always export what is visible here. So if we subset our view here by individual projects or by individual groups, then it will only export the selection here. And we just need to click the export button, and this allows us then to download a CSV file. And I've opened now the CSV file in Excel, and as you can see, it's containing all the columns and metadata that were part of the individual data sets here. And like this, we have a nice overview that we can share on other ways and not through the ReadStore app. For instance, we have a group here of all the tissues, normal and tumor, and the replicate information. In a similar way, we can also export projects. We can also export the FASTQ files that make up the data sets, and like this have a flexible inventory of all our data. So let's look at the download function. With the download function, we can get access to our FASTQ files and attachments. For this, the download functions are usually available as part of the detail view. So we can navigate here while clicking on while selecting a project, we get access to the detail view. And if we select attachments, we get a list of all the attachments here, and we can select the attachment that we want to download and then click the download button, and we will be able to get access to this attachment PowerPoint presentation here. We can also, in a similar way, download FASTQ files directly from the browser. One disclaimer here: if the FASTQ files are very large, then this will not work, and then you should maybe use the command line interface that is not limited in the size to download FASTQ files in case you need them. For smaller FASTQ files this works though. We can just select the read here, then scroll down and click the download FASTQ button, and this will start the download or give us a warning if the file is larger than 5 GB. And finally, we want to have a look how we can delete data sets. For this, we again go in the update view. And in this dialog, we have the trash bin here that says delete project. And we can confirm the delete. And like this, we can get rid of projects. This will not delete the data sets in the projects, just disconnect them. We see that the datasets are now not having any projects attached anymore. And similarly, we can also select an individual datasets, go to the update and delete the dataset. If we want to delete attachments, we can go to the attachments tab and delete the attachments in there. And those are the main project dataset management features. If you have comments or questions, please leave them here or reach out. If you want to use ReadStore, you will find more information in the video notes, and I wish you a great day.